Great. We are going to get going. I'm so happy to see so many of you here. And it's really my pleasure to introduce the seminar speaker today, Dr. Diana Valverde Mendez. I'm sure I mispronounced your name. But I have recently learned over the past summer that most Hispanic names are threefold. Your name, father's side, mother's side, or something. So we work out the whole permutation after a few generations. Somehow the grandmother's name get dropped, which was still a little bit upsetting. Anyway, Diana and I met a couple of years ago when we both served as volunteers at the APS D-Bio desk at APS March meeting. So, it's really nice. You just sit on the hallway. There's a nice desk. You try to encourage other people to become your community members. And then there's a lot of times we end up chatting about sort of career paths and things. It's really a fascinating story. I don't have a lot of time to tell you, but basically, Diana grew up in Costa Rica, a beautiful country I have not visited yet. But you went, I'm sure you've been to places. The people there probably love that too. So anyway, and she was saying that, when you grow up in a small country like that, you just have to convince people that you know your physics. As undergraduate, she actually did electrical engineering, but with a scholarship, she, spent the money and actually flew in the States and visiting several universities, and then ended up doing a physics PhD at Princeton University, and working with Josh Chavez, and became sort of a prominent professional biophysicist. And with her degree in physics, I guess the physics side is well under control. But I know the person she's doing postdoc with, Professor Jin Yan, who is actually on the faculty in molecular biology, even though he did his PhD in soft matter physics. So really fascinating story. So Diana is going to tell you about her PhD work in Princeton. Well, she's probably also going to touch upon some postdoc work she's currently working on, which I found fascinating too, on biofilms and stuff. Without further ado, I remember there's a longer title in the thing, but this is actually good. 3D diffusion in live E. coli cells. Go ahead, Diana.
Thank you. Thank you, Jane, for the really nice introduction. So I'm just going to get started. The first question that you might be asking is, why even look at bacterial cells? Why look at diffusion inside bacterial cells? And when you go to a microbiology textbook, this is the cartoon of a bacterial cell that you get. It's pretty sparse, right? There seems to be some DNA, maybe some additional DNA over here, and a few ribosomes. And it seems to be a pretty boring system to look at. But in reality, if you were able to zoom in into a little part of the cell, what you would see would be something more like this. It's incredibly crowded. To give you an idea of how crowded it is, the volume fraction is about 30%. But not only is it incredibly crowded, hopefully you can see that there are a bunch of different particles of different sizes. And it's so much so that depending on how you define it, there's 4 to 6 orders of magnitude of components of size. But there's not only polydispersity in terms of size, but also charge. So there's about 3 orders of magnitude of different charges. And what's really interesting is that this charge has a bit of a hierarchy. So small things, like ions, tend to be positive, whereas large things, such as ribosomes in the DNA, are very negative. The whole cell is very close to neutral. It's a little bit negative. But there is this hierarchy in size of charge that is not fully well understood. And we will see later on during the talk that maybe there is a reason for this. Additionally, to make things more interesting, the bacterial DNA is about 1,000 times longer than the bacterial cell. And yet it's compacted in this really small structure called a nucleoid that occupies between 25 and 75% of the cell. And this structure is not bound by a membrane. And yet, it's been observed that ribosomes that are not able to penetrate into this DNA-rich area. And this is understood to be due to entropic reasons. So hopefully this has convinced you that looking at how things move inside of a bacterial cell is interesting because there is an incredible amount of complexity in the bacterial cytoplasm. And I'm not the only one who thinks about the things that this is interesting because over the past 20 years, people have gone and tried to see how do things move inside bacterial cells. And from these kind of experiments, we know a few things. We know that things move a little bit slower than they should just by normal diffusion. And we know that this the slowdown seems to be particle size dependent. Additionally, we know that there are weird things happening. There has been some jamming transitions that seem to have been observed, and some people have called the cytoplasm glass. And so, but no satisfactory physical mechanism or reason for why things are the way they are have been provided. And so we wanted to build a systematic approach to really go and see what is happening with diffusion inside bacterial cells. So we identified two major limitations that experiments before ours had encountered. The first one was that many of the tracer molecules that people use, so you have a fluorescent particle, you put it inside of your cell somehow, and you see how it moves inside of the cell. And based on that trajectory, you can extract some mathematical measurements that you can use to extrapolate what kind of motion you have inside of the cell. But those tracer molecules that people were using were not ideal. Sometimes they would assemble into different sizes. There was no control over size many times. Parameters as important to us, like charge, that maybe a physicist would think is super important, had not been considered at all. And additionally, sometimes the particles that were used were from the bacteria. And so you couldn't separate biology from physics. And so for that, we developed genetically encoded multimeric nanoparticles for bacterial cells. These are probes that have a tunable size and charge. They were originally developed at the Holt lab at NYU for research in eukaryote cells. And working in collaboration with them, I made them for bacterial cells. And the way that they work is that you have a scaffold protein that likes to bind to itself in fixed numbers. So 180 or 240 of these molecules will come together. And it always does the same thing, and it only depends on what scaffold protein it is. And it will form a little icosahedral ball. And what is nice is that if you link a fluorescent protein to that scaffold, then what you will end up is with a little ball that is completely decorated by those fluorescent proteins. Meaning that all of the interactions that that particle will have with the cell happen through those fluorophores. By changing the scaffold protein, we can change the size of the particle, and by changing the sequence of amino acids that makes up your fluorescent protein, you can actually change the charge of the particle. And so in this project, I'll tell you about 3 different sizes, 20, 40, and 50 nanometers, and 3 different charges. One that is very negative, one that is close to neutral, close, and another that is positive, very positive, okay? And you might be wondering, well, you told me that there is like 6 orders of magnitude of size inside of that cell, so where are we with these particles? Well, we're about the size of ribosomes and polysomes, which are very important molecules inside the cell, so there is some relevance in biology. And when we started this project, there was really not that much about this length scale that had been explored, which is kind of like the mesoscale. And the ribosomes have a charge of about minus 3,000 electrons, so they're pretty comparable to this 40 nanometer particle that we have. Okay. The second limitation that we observed is that all of these experiments that I mentioned before were done in 2 dimensions. And when you have motion that is inherently 3 dimensional, and you project it down to 2 dimensions, you're bound to lose a lot of information. So I want you to think of a sphere. Think of a particle diffusing in the surface of that sphere. If you were to see now the projection on a circle, it would be impossible for you to tell that that particle is moving in the surface of the sphere. And so this is the kind of measurements that have been done before until we started with our work. And so for our 3D tracking, we use biplane microscopy. And the way that biplane microscopy works is that in traditional microscopes, you have all of your optics, and in the end, you have a lens. And that lens focuses down your light into the camera. But we have an additional module called biplane module that is super simple. It just consists of a beam splitter and a mirror. And this beam splitter is such that half of the light goes straight through and focuses in the camera normally, as it would always do. And the other half of the light will bounce off the beam splitter at 90 degrees, reflect off the additional mirror, and then go into the camera. But now you see that this light has traveled a longer optical distance. And therefore, the image that will form in the camera would be at a different focal length. And so here I'm going to show you a C-stack of beads. So we're just sweeping along the optical axis. And as you can see, first the beads come into focus here, they're defocused in the bottom, and then they focus in the bottom, and they're defocused at the top. So simultaneously, we get the information from two focal planes. And I will show you why this is important. So imagine when you have a sample of point particles in your microscope, the light from these point particles will go through your optics, and it will diffract. And because there is finite size apertures in that microscope, you will lose all the high frequency components of that light. And so the resulting image of these point particles is actually a blurred out image called the point spread function. And so whatever you image in a microscope is a convolution of your sample with that point spread function. And as you can see, that point spread function makes you a bit like a circle in XY, and then it makes you like this elongated image in Z. Okay? So now imagine that I take my microscope that only has one plane, and I focus it at the particle. And then I know let's say that I know what my focus is. The next time step, the particle might have moved up or down. But because this point spread function is symmetric, it's not possible to tell whether the particle moved up or down, right? And so I'm not able to determine what the position of the particle is. But if we now have instead two planes, then when we say we're in focus, we're not actually in focus in either plane. We're more or less straddling that focus point. But we can extrapolate what is the position of our particle that it's at focus. Now, the particle, imagine that these are my two planes. The particle moves down. Then, when the particle moves down, the top plane will now be more defocused, and the bottom plane will be more in focus. And so now I can tell for sure that the particle moved down. Same thing if the particle moved up, now the top plane will be in focus, and the bottom plane will be defocused. And with that, I can tell that the particle moved up. So this is the principle by which, with only having two planes, you can fully extract the C position of the particle. And
sorry, quick question. How do you keep these two images separate?
Yeah, so what you do is that in the illumination side, you use a mask, and we use that little rectangular mask, such that you just have like a smaller, you calibrate it such that the rectangular mask maps to the top side of half of the camera and the bottom half. Okay. So it's like you mask the illumination to fit into your camera chip. Okay. And then finally, what you do is that you will be able to obtain like the image of your sample, and from that you can extract this pixel intensity, and then you will be able to fit a Gaussian, and with that you find out what is the position in each plane. So here you have top and bottom, top and bottom, top and bottom, and you can tell where is that position. And then if you do some clever summing with a weighted sum, then you can end up with what is the position of the particle in Z. And so here you can see a trajectory of a typical particle. Okay, so this is what our data looks like. We have our DNA labeled in red, and our particle in green, and we had an additional membrane stain in blue that I'm not showing here. And as you can see, the particle is moving between the two planes, so sometimes it's focused in the top, sometimes it's focused at the bottom. And from this, we are able to extract and reconstruct a full 3D trajectory of what that particle is doing in space. And in traditional 2D tracking, what you would see would be this projection here in red. So again, it's like that mental example I made you of a particle diffusing in the surface of a sphere. When you project that down to 2 dimensions, it's really hard to tell what is going on. But if instead you focus in this back projection here, and I have my bottle to illustrate it, that's just like looking directly down the axis of the bottle, then what you see is that that particle is actually fully excluded from the DNA area of the cell. So it's exactly that example I just gave you with the sphere, but for a cylinder. So this is a kind of really powerful information that you can obtain from three-dimensional tracking that is not accessible in 2D. Okay? So I think this is a good place to stop and ask questions. Yeah.
Okay, so simple question. So the thickness of the E. coli is comparable to the two different focal planes.
Yeah.
And, okay. And so when you did the can thing, that was a great prop. When you did the thing with can, yeah, thank you. You're not going to be able to track a particle completely in 3D if you use the same plane separation you did for the. So the two planes are actually comparable to 500 nanometers apart, I guess.
Yeah. So the two planes are like here.
Yeah.
And the two planes are actually separated by like 200 nanometers optically.
Okay.
But that gives you a working range of about 1.5 microns, which is the size of the cell.
Oh, it gives you 1.5 microns.
Yeah, so
okay.
When you go back to this, if the particle is visible in either one of the planes, when you do the weighted sum, it tells you where the position is. So you have to calibrate it for your system. But in our case,
so in the width of the Gaussian is what? Like in the z direction?
The width of the Gaussian is about 400 nanometers. It's
Okay, so that's bigger than. All right, thank you.
Yeah.
For real-time tracking on, in terms of the question I want to ask is in terms of the position accuracy. So I understand that from the Gaussian fit, you can get the position much better than the resolution, the diffraction limit.
Yeah.
But you needed to have enough photons to do that, right?
Yes.
So this thing is moving so fast, and how can you be sure you have enough photons to do this fit?
Yeah, so we're very lucky that these particles are about 180 fluorophores, so they're pretty bright. There's people who are working with 1 fluorophore, and in that case, they have to worry a lot about whether or not they have enough signal. But in our case, because we have so many fluorophores.
And then this fluorophore itself is compact, the structure-wise, it's not floppy. How big is it?
It's about 4 nanometers.
Oh, okay. Okay, so that pretty much sets your limit.
Yeah.
Okay, so what's the resolution?
So our resolution is about 30 nanometers in each direction.
Okay.
And normally for these really bright particles, if you only had one plane, your resolution in XY would be like 1 nanometer. But because we're mapping those two planes to create our full 3D information, we are accounting that as part of our localization error because the mapping between the two has like 30 nanometers of error. And that's something that I forgot to mention, but when you have only one plane, you can see that particle went out of focus pretty quickly, right? And so one of the things that we get out of having biplane is that we can track our particles for really, really long times. So we're basically limited by how long it takes for the fluorophore to die because we never lose it. And so this kind of temporal resolution was not a thing that existed before these experiments. And it's what allowed us to actually get at the root of what is the physics going on inside of the cell, because we had long enough trajectories to explore this.
How do you make sure there's only one inside the cell?
Yeah, so if you just tune your biology properly, don't induce the plasmid we were talking about earlier, then every now and then you get one. And you just choose the cells that have one. Yeah.
Yeah, I got an uninteresting question. How do you get them into the cell?
Yeah,
like experimentally?
So what you do is that you introduce into the cell a plasmid, which is a little like circular chunk of DNA.
And how do you do that? You just shake it up with the cells?
You electrocute the cells, or you expose them to some chemical treatments or something like that, and the cells will be willing to take up all the DNA.
Okay.
And then once the DNA is inside of the cell will make the proteins, and the proteins will self-assemble inside of the cell.
So interesting. Okay.
Willing is generous.
Well,
Sorry.
there are some cells that willingly do it without you having to electrocute them
You can probably kill like 1,000 before you get one that works.
That's fine.
I've killed a lot of bacteria in my Okay, cool. So now that we know what the technique is, I'm going to start telling you a little bit about the results. So here I'm showing you a 2D histogram of all the localizations in that cell. And so in all of the cells. So I'm just pinning over all of the localizations. And so what you can see is that for the 20 nanometer particles, most of them are localized in that DNA-rich area in the nucleus. Whereas the 50 nanometer particles tend to be localized in the periphery of that DNA area. And you can see that very well here in the cross-section. Whereas the 40 nanometer particles, whilst they're a little bit excluded from the DNA, they kind of are able to go everywhere inside the cell. So there is a clear dependence on your particle size to where you localize preferentially inside the cell. We can go a step further and plot the probability, the radial probability distribution for these particles. So we take only the cylinder part of the cell, and we find the radial position of each of these particles, and then we created this density distributions. And as you can see, the probability distribution shifts towards the cellular periphery as the particle size increases. And this result here for the 50 nanometer particles was completely expected. I told you before that this happens for ribosomes, for entropic reasons. But this 20 nanometer result, at the beginning, we were pretty surprised because it hadn't really been seen before. And we hypothesized that maybe what was happening is that these same entropic reasons were causing the small particles to go inside of the nucleus because outside it's so crowded. And so to test this hypothesis, we teamed up with the CL lab at Stanford. So this is a theory group that they make these beautiful colloidal simulations where they model all of the where Al Sunol, who was a grad student in the lab at the time, now he's a postdoc at Harvard, went on and created this model of the cell where he modeled all of the important cellular components and their hydrodynamics, their electrostatics, and their thermal interactions. And so the first step was to see how good of a representation is this cell to our actual physical cell. And so for that, we look at the percentage of nucleoid occupation time. And this is a measurement of how much time do you spend in the nucleoid versus your full length trajectory. And we see really good agreement between experiment and simulation. There might be a little bit of a discrepancy, a tiny discrepancy for the 20 nanometer particles, but considering we have a system that is basically a sphere and another that is a cylinder, and that he's done all of the simplifications, it's pretty remarkable that we got such good agreement. So trusting that the simulations give us a good picture of what is happening inside the cell, Al went and measured the different void sizes, so the little pockets in the nucleus. He measured the size distribution of those. And what he saw is that about 90% of those pockets in that nucleus are larger than a 20 nanometer particle, meaning that a 20 nanometer particle can get into the nucleus, to much of the nucleus. Whereas for a 50 nanometer particle, only 52% of the voids are accessible. So what we think is happening here is that the large particles, such as ribosomes and clumps of ribosomes and all of this, are excluded by that DNA because they cannot get in because of the pore size. And then, because all of these particles are outside, this actually raises the volume fraction outside about 3% larger than inside, which means that if you are able to go inside, you'll be pushed inside by a kind of osmotic force. And so that's what's happening here. So that's why the 20 nanometer particles are enriched in the nucleus. Yes.
I just have a question for, to what role do the electrostatic interactions have in this distribution that you see?
Next slide.
Oh.
Okay.
Getting ahead of myself then.
Yeah. Yeah, so now that we know what happens with size, we saw that our 40 nanometer particles kind of are able to explore the full cell. So it makes sense for us to see what happens if we change their charge. So we fix the size at 40 nanometers. And now I remind you, we have a really negative one, a really positive one, and this kind of neutral one I told you about, it really is kind of neutral when we look at our simulation results. Like the physics looks pretty similar. So let's think of this negative, positive, and neutral. Okay? And what we first see is that the negative particles tend to be enriched inside of the DNA, and the positive particles are pretty excluded from that DNA area. And again, the neutral particles kind of can go everywhere. We can look again at our probability distributions, and we see that enrichment in the nucleoid. But maybe for the positive particles, we don't see that enrichment so much. But I told you that before that, I'm just looking at the cylinder part of this sphere cylinder when I'm mapping this, when I'm making these probability distributions. But you see that lots of particles are in the poles. So if I instead plot the percentage of particles found at the poles, we see that there's a huge enrichment in the poles. So about 25% of the positive particles are found at the poles. And just for reference, 15% is what you expect just by a uniform distribution in a sphere cylinder. So 15% is what you expect, 25% is quite a lot of enrichment. So it looks like based on size, the more, as you go from negative to positive, you're expelled from the nucleus.
That's very counterintuitive too, right? You
Yes.
would think positive charged ones will want to be mixing with DNAs, but
Exactly.
But
So
the bilength is extremely small, I assume.
the what?
The bilength.
Yeah, because there's
So
so
the
much
charge
ion
is all
screening
screened,
and all of that.
basically.
Yeah. So
Sorry, Diane, on the last slide, your x-axis, what was that? Was that the charge or?
yeah, this is just a particle charge.
So that charge doesn't.
Oh,
really
it says
move.
diameter. Oh, I'm sorry. Oh,
Okay,
I have to.
but also
fix
the
that.
charges are different, right, from the charges you have. So I was just wondering.
Oh, okay, yeah.
They don't feel
So,
the charge until
During revision, we realized there was a problem with the calculation of the particle charge,
Ah,
and so we went ahead and we changed everything, redid the simulations, and I forgot to update this figure. I'm sorry about that.
Okay, no worries.
Yeah. But all the results remained the same. It's just during revision we noticed that there was this small error.
Okay.
Yeah. Sorry about that. Oh, good catch. Okay. So now we go back to our correct particle charges, and then so here I'm showing you again the percentage of nuclear aggregation, and I think that it's really interesting that although the two negative particles have very different distributions, they have a similar aggregation level in the nucleus. And then once again, you see that the positive particles are very excluded from that nucleus. And as Jay said, this was actually super counterintuitive to us because we know that the DNA is really negative. And so we said, like, what is going on? This doesn't really make sense. Until we realized that ribosomes are even more negative, and there's many of them. And so what Al did was that he went and he measured the coordination number, and that's just how many of a particular particle are found around your particle. So that sounded weird. So for example, how many positive Crowders are found around the sphere? And this is normalized to like no charge. And what we see is that as particle charge goes towards positive, then there's more and more ribosomes surrounding that particle. And same with DNA, and it's the opposite for positive and negative Crowders. So this plot is a little bit hard to follow, but if we instead look just at a snapshot of the simulation, it becomes clear that our negative particles are very transiently interacting with these small positive things. Remember that all the positive things in the cell are really small. So they come, they interact, but they cannot affect that motion all that much. Whereas the positive particles are fully surrounded by those ribosomes. The ribosomes, again, are very negative, but they're also very motile. So sometimes in the simulations, we see that the particle is next to the DNA, but even when it's next to the DNA, it's completely surrounded everywhere else by ribosomes. Because the DNA is not moving fast enough, so it cannot arrange itself around the gem, whereas the ribosomes that are free to move are able to constantly be surrounding that gem. And so effectively, they make the particle grow in size, and now it can really not go into that DNA rich area. So that is the reason why the positive particles are excluded, and the negative particles are a little bit enriched in the middle of the cell. Does that make sense? Great. Okay, any questions?
I just want to make sure I'm understanding it right.
Yeah.
So the positive gems attract the negative ribosomes, and that increases the effective radius, preventing them from getting in.
That's correct.
So what is the effective radius of the gems that have kind of gone covered with these ribosomes?
Probably around like 80 nanometers.
Okay.
Yeah, so much bigger than even our 50 nanometers, which is
So
why
it doubles basically.
Yeah. Because each ribosome is about 20 nanometers, so if we just assume that there's like the gem and 2 ribosomes, that's 80. Sometimes the ribosomes have chains of ribosomes, so it could be even larger.
Cool.
So ribosomes are negatively charged intuitively because you don't want them to be stuck with DNA. Is that why they are?
Yeah, so it seems to be that this and combined with the next slide, where we see that mean square displacement is a measure of how far do you move in a particular amount of time. And so this is just saying that positive things move really, really slowly. And hopefully you can see that in the simulations. And then very negative things move slower than neutral things. So charge slows you down. Positive charge would slow you down to the point where you cannot do anything. You're stuck in a corner, so fully surrounded by ribosomes. So I think that this is why there is this size charge hierarchy inside of the cell. Because if you had large macromolecules of positive charge, they wouldn't be able to have any function in the cell. And so they would get lost by evolution. It could be that if we restarted the universe, it would be the other way around, and the large things were positive and the small things were negative. But it does seem like it's necessary to have this hierarchy. Yes?
So going back to, I have a couple of questions. A quick question. Is there's some kind of a membrane around the nucleoid? Is that
No, not at all.
So this is just phase separation?
Yeah, entropic separation, yeah.
Okay. It's a very, I don't know how to think about that. And then the ratios of the diffusion constants. So Severia asked that question about, getting dressed by the ribosomes so that you're bigger. I mean, so quantitatively, do you see like, what, it's supposed to go like the radius or something, or 1 over the radius?
Yeah.
So does that work?
It kind of works. It is like the slowdown is consistent. It's not exactly like half or something,
Okay.
but because like the number of ribosomes is variable.
I see.
But yeah,
I see.
it is of the right magnitude. Yeah.
Thanks.
Diana, your data, does this come all from one cell? Like one E. coli, or did you average
No, we
many?
have a bunch of different cells. And then,
And you averaged everything over many, many different cells?
yes.
Okay.
What about slope? Like what
Next
slope?
slide.
I can't wait.
This is great. You guys are really following the storyline. Okay, so now we can go and look closer at our mean square displacement. So I very briefly said mean square displacement is a measurement of how far on average do you move squared. And so what you do is that you look at different time steps. So maybe one time step, you look at the displacement of all of your particles in that time step, you average, and that's this first data point. Then you look at 2 time steps, do the same thing, and that for all of your time steps. So here's where I want to point to you that we actually do have really long trajectories, because you can only get so many different orders of magnitude in time unless you have really long trajectories. So this is where having the biplane microscopy really helps us. Okay, so mean square displacement is proportional to time to an exponent. If that exponent is equal to 1, then you have Brownian motion. If that exponent is smaller than 1, then it means that you're moving less distance in the same amount of time, which means that you're sub-diffusive. And why could you be sub-diffusive? There's a few reasons. It could be that you get trapped in a little pocket of something. It could be that you're binding and unbinding from something. It could be that you're just finding a wall, and that wall is not allowing you to move. So if you notice, if we get the log at each side, then this alpha actually becomes the slope. So in a log-log plot, the alpha will be given by the slope of these curves. And so you can see here that all of these curves are sub-diffusive. Okay. Additionally, you could see that at really long timescales, we're seeing that all of the curves are platonic. But this is completely expected, because we have long enough sampling for long enough that we're actually seeing that you can't move past the size of your cell. And you see that the 20 nanometer particles are confined to a smaller region because they are mostly localized in the nuclei. So the bigger particles are seeing the whole cell, and the smaller particles are mostly just seeing it. Also notice something that goes back to the question before, that we have the yellow curve is the 20 nanometers. And from Einstein's diffusion coefficient, we would expect our 20 nanometer particles to be moving twice as fast as our 40 nanometers. But because they're confined to that nucleus, they're actually effectively being able to move less. And so our 20 nanometer particles are slowed down just by being confined to that nucleus. Okay? So what is the origin of this sub-diffusion? Is there really some interesting new physics, or what is going on? Well, if you go now to the simulations, back to the colloidal model, at really small timescales, like picoseconds, you see that all the exponents are really close to 1. And so this tells you that it's not the hydrodynamics, the electrostatics, or the thermal interactions that are causing these particles to be sub-diffusive, because we're not seeing that slowdown at short timescales. So in order to bridge the gap between our simulations and our experimental timescales, we used random walk simulations. And in this random walk simulations, you instantiate your particle pretty much anywhere in the cell, and you give it, and you allow it to move inside the cell by drawing displacements from a normal distribution. So just Brownian motion. But you don't allow it to go past the edges of the cell, and additionally, you give it a probability of tunneling into the nucleus, given by that occupation fraction that we measured earlier in the experiments. And when you do that, you see that there's a really good overlap between our experimental data and the results of the simulation. So you run the simulation many, many times, and then you average over all of those trajectories, and you get the mean square displacement. And you see that there's almost perfect agreement. So this is suggesting that what is going on is that it's actually all just a result of the geometry in which the particles are moving.
So this must be, people must have seen that if you make a 40 nanometer particle in a half a micron microfluidic cell, and you do an image of that, you should see the same thing, right?
Yeah.
People must have done this.
I don't
Except
think
half micron is very, very small, too small to have, traditional microfluidic chamber. No, half micron is extremely difficult. When people do microfluidic, maybe they do a few microns or something. Half micron, you need special optical.
It's just even lithography, right?
You can produce it, but
Yeah.
to study
Okay.
particles in it.
All right, okay. So people just didn't bother to do it, but this should be an easy experiment.
Yeah, you would think.
It's hard.
There's also there's also the little thing about having a porous nucleoid in the middle, because that is also affecting the diffusion. But we can go a step further. Yeah.
I just have one question about the simulation.
Yeah.
So the particle could be initialized at any point in the cell. From just on the biology side of things, do the GEMs get produced out on the outskirts with all the ribosomes?
I think so, because the proteins are made there.
So there's no way for them to ever start in the nucleoid, right?
Probably not. But
okay. It doesn't seem to be affecting your model though.
Okay, so I made the first version of the simulations, and then my collaborator did the second version. So I don't actually know if the ones in the model, they could start anywhere. I think in my version they could only start in the first, but I don't think it matters all that much.
Yeah.
I'm pretty sure the porous media people know about this a long time ago.
Yeah, I think especially the size filtering, that's something that is even used to separate particles of different sizes in chemical labs and stuff. So it is definitely something that makes sense.
Yeah, the people like in Schlumberger, they used to study things in porous media diffusion, in porous media, the oil people, right? They try the exxon people. They do this all the time.
Yeah.
But intuitively, I still don't get it. Why, with 20 nanometer cavities, right? If they can't get in, why do they prefer to be in there?
It's because the volume fraction outside is bigger. So it's similar to when you have depletion aggregation, where you have two things, and if nothing can get in, they kind of gets pushed together, and that's called like an osmotic pressure. Those 20 nanometer particles are feeling an osmotic pressure because there is more stuff outside, so they can't get between two ribosomes.
Okay.
But so the ribosomes come together and they push the particles into the. Yeah. More questions? Okay. So we can go a step further and we can say, what happens if we just normalize all of our mean square displacements by the diffusion coefficient? So with that, we're getting rid of like the speed, and we're just keeping the nature of that diffusive process. And as you can see, for all our particle size, big particle sizes, they all collapse on top of each other. And our smaller particles actually end up different. And if we now overlap the results from our random walk simulations, we see that the results for the large particles are very well recapitulated by a porous spherical cylindrical shell, and the results for the small particles are very well recapitulated by a nuclear confinement, so being confined to the center of the cell. So what this tells us is that a lot of these crazy observations of sub-diffusion, anomalous diffusion inside bacterial cells, and all of these things were actually a result of the geometry of the system and not so much of new interesting physics.
No biology. Whatever we just want.
Finally, you might be asking that question: are these even good probes to measure biological systems? Do they recapitulate what you expect from a ribosome? So here we have the mean square displacement, the 2D mean square displacement for ribosomes that someone had gone and measured, and then we overlaid our 2D. I made like a 2D MSC, and I overlaid our results, and you can see that there is really good agreement between our tracer particles and those ribosomes. And in particular, our 50 nanometer particles that are negative and large seem to be behaving very similar to the ribosomes that are negative and large. So our bGEMs are really good model particles to study what is actually happening with real components inside of the cell. Okay, so with that, I want to give you some take-home messages. Particles localize inside the cell based on their size and charge, and depending on where they localize, will determine their apparent diffusion. And the small particles seem to be diffused slowly because they're confined to a smaller region, whereas charge slows all particles down. And finally, random walk and colloidal simulations indicate that a lot of this weird anomalous diffusion that had been observed is just the result of diffusion under confinement. But I do want to make a note here that just because it is normal diffusion, it doesn't mean that the cell actually cannot leverage this to achieve function, right? Because for reactions that are happening in the picosecond scale, this is all moving at what you would expect from diffusion. But for certain capture processes inside the cell, if two things are made close to each other, they will remain closer for longer. If two things shouldn't be close, they can be made in different parts of the cell, and they will probably not find each other. You can keep ribosomes localized near the center of the cell where they can go and start finding those nascent mRNAs better. And so I find it really remarkable how biology can leverage physics to achieve function without having to do any energy expenditure. Because in bacterial cells, there is no active transport motors. There's nothing moving things around. So all of the function that has to happen inside the cell has to happen just by leveraging physics. So I think that this is pretty remarkable. Okay, so with that, I'm going to thank my collaborators, thank you for listening, and thank the funding sources. And if you have any questions, we have a we have a preprint out in the archive. This is a QR code. We should be uploading a new updated version with the revisions. It's currently under revision at PNAS. Hopefully it will get accepted soon. Okay.
Can I return to this question about what's keeping the nucleoid? What are you calling the nucleoid if there's no membrane? What does that mean? And do your results start to give you some ideas as to how the segregation that makes some?
Yeah. So what makes the DNA be compacted in the center of the cell is an open research question. We know that there is some part that is like entropic reasons. The ribosomes have more degrees of freedom if they're not mixed with the DNA. We know that there's an electrostatic reason. The DNA is negative, the ribosomes are negative, so they're pushing each other apart. But if you take all of those effects into consideration, it still doesn't add up. So there's also like some linking between the different parts of the DNA. There's like some coiling in the DNA, and all these things are still like actively being researched. It is worth pointing out that at different, if the DNA, due to nutrient conditions or something else, has different pore sizes, then you would expect diffusion to be different. So another interesting research question that could follow is, are there different conditions under which the diffusion is different inside the cell, and how is the cell changing like its physical characteristics to achieve different functions? Yeah.
So kind of throughout all of your your simulations and experiments, you assume that the nucleoid does not move and it stays in the center. So is that kind of like does that accurately happen in realistic cells?
Yeah. So for these experiments, we mostly work with the stationary phase cells, which are cells that are basically cells grow, divide at exponential rate, and when they run out of nutrients, they go into the stationary phase. They're not dead; they're just like not actively growing as much. So we chose to work in this condition because the nucleoid itself is not changing. During exponential phase, the nucleoid is constantly changing its shape and stuff, and that would make it more challenging to do this research. But in the simulations, we also tried like a static nucleoid or a nucleoid that is moving and stuff like that, and the results are pretty much the same. Yes.
Yeah, you have this remarkable spatial resolution and time resolution, and you seem to be having collected a lot of data there. So I was just wondering, like, can't you in your analysis like separate like the times and moments when it's inside the nucleoid and outside, and analyze these separately?
Yeah.
You should be getting very drastic differences in these cases. But maybe they don't stay there too long, or
I tried, but the data was a bit too noisy for us to feel like it was like good enough to go into the paper. We do have some like figures from simulations where our collaborators did look at, are there different exponents inside, outside the nucleoid, and all these things.
Yeah.
And it does seem like it's a little bit more sub-diffusive in the nucleoid as you would expect because of that